body here, and I certainly said it's an oncogene, but in tumor suppressor, it's wrong, both are for tumor suppressor genes. The CpG island gets methylated and the gene body gets unmethylated, leading to lower expression of the tumor suppressor gene, and that is of course causing the disease. And then someone came to ask, can arginines also be acylated? No, they cannot. It's the lysine that is acylated. On arginine you can have acetylation, no acetylation but methylation. So good that you keep me short, because while I'm talking I said something wrong. It was correct on my slides, but wrong while I was saying it. After the break now we are going to talk about genetic imprinting. And genetic imprinting are is the underlying mechanism for many effects that we see when we make crosses, artificial crosses, let's say, between, for example, what you see here, a horse and a donkey. And this imprinting, the imprinting is an epigenetic mechanism that underlies, that explains why we see differences when we do reciprocal crosses. Like here on the left you see the hinny, which is a female donkey with a male horse. And on the right you see a mule, which is a female horse, and a father that is a donkey. And as you can see here, so in Dutch that's male dier and male ezel. What you see here, I think, is that the children, so the offspring looks a bit more like the father. This has a bit more, certainly the head here is a bit more like a horse, while here the head really looks like a donkey. So you see some things that are quite different between in which direction you make the cross, and that is influenced by patterns that we call imprinting patterns. For this cross, it's not that well studied. So for this cross, I don't have the real mechanism. I don't know the real mechanism. I looked it up. I couldn't find it, so it's not that well studied. But for some other ones, we do know. So another one that is a cross that has been done in zoos is the cross between a lion and a tiger. Again, in both directions. And on the left side, you see what is called the offspring is called a liger, when you cross a female tiger with a male lion. I hope that you see that this is a huge animal. It's a really incredibly large animal. Way larger than a regular lion. Massive. While here, in the other direction, a female lion crossed with a male tiger, you get, in fact, a quite small animal. The tiger is relatively small. So, bred in zoos, but sterile. They cannot reproduce further. But these crosses are possible, and the fact that you see differences between them, whether for the reciprocal crosses, is explained by imprinting. So here again shown, a male lion crossed with a female tiger, you will get a huge animal. The liger is more than 400 kilograms, weighs more than 400 kilograms. While the tiger here is way smaller, typically even almost the same size as this female lion, or smaller than the parents. So what explains those kind of differences? That's the mechanism that we call parental genetic imprinting. And parental genetic imprinting is a monoallelic gene expression determined by the parental origin. So I hope you remember that both mammals are diploids. We have 2 alleles for every gene, and usually both alleles are active. They are expressed. But 1% of our genome is not biologically expressed. 1% of the human genes shows parental genetic imprinting. One of both alleles is active, the other one is inactive. So for example, a very, very, very well studied example, IGF-2 is one of those imprinted genes. And in IGF-2, only your paternal copy is active. The maternal copy of the paternal allele of IGF-2 is silenced. So under normal conditions, as all of us, I assume, we are all having, being normal human beings, IGF-2, only your paternal allele is expressed. The maternal copy is inactive, or we also say that it's imprinted. However, imprinting can also be tissue specific or time dependent. So it could also be that it's for some other genes. For IGF-2, that's not the case, but for other genes, it can be also only in specific tissues or only in specific times during development that imprinting is extremely powerful. Now, parental imprinting has already been discovered in the 1980s, and I'm sorry, this is a bit cruel. These experiments that were done in the 1980s, they made uniparental mice. So they made embryos from just transplantation of pronuclei in the zygotes. So, for example, they made a zygote out of 2 female nuclei, or zygotes out of 2 male pronuclei. And so that was possible by transplantation, and when they let this embryo develop further, compared to a regular embryo, they saw, of course, not very surprisingly, they saw some weird things happening. And they saw the embryo is not developing well. However, it depended on in which, if they used 2 female nuclei or 2 male nuclei, you had a different result. When they worked with 2 female nuclei to create a zygote, they got a relatively normal embryo, looking quite normal but very small. But when they then looked at the placenta, they saw that placenta was extremely small, so undersized. However, when they worked with 2 male nuclei, they saw, in fact, something very spectacular, a huge placenta. The embryo didn't develop well, so that's this one here, a tiny embryo that, in fact, doesn't really look like an embryo. So, of course, you get abortion then, and this doesn't develop further. You cannot grow real mice from it. They could only study it at the embryonic phase. You will never get, you will not get further, of course, because of that abnormal development of the embryo. And the evolutionary theory that explains imprinting is, in fact, the theory of the conflict between sexes. There is a conflict there of what, as father or as a mother of this embryo, you want to achieve. Many of the imprinted genes are having specific effects, are related to placental growth, embryonic growth, and so on. The bigger the placenta, the better the embryo will be nourished. So when a placenta is huge, as mother, all your nutrients are driven towards that placenta going to that embryo. So that's good for the nourishment of that specific child. As a father, that is good. You want your baby to grow good. It's your baby, you want it to grow well. You don't, as father, and now I'm saying it a bit cruel, as a father, you don't care what happens to that mother or what happens to the next children. Because those other children could be from another father. You want your child now to grow well. So it's very important that your child gets well nourished. So paternal expressed genes, so alleles that are from the father and that highly expressed are typically promoting placental growth. Maternally expressed genes, however, say, whoa, 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 whoa. Stop. Don't do that, because I need to survive as well as a mother, and maybe I want more babies. Maybe I will get more offspring later. So, the mother tries to protect herself and next offspring potentially from being drained out, let's say, from all the nutrition. So there is a conflict there between what the father wants and what the mother wants for the other. And for many of the imprinted genes, we see indeed that pattern, that they are following this, that they are indeed promoting placental growth, or on the other side, as a maternal expressed genes, mainly counteracting. And what is even more remarkable, I think, is that we only see these imprinting patterns in animals that have a placenta, or in plants which have something like a placenta at the end of birth. So only in those organisms imprinting is observed. Now, how can you get monoallelic gene expression? How is it possible that one allele is active and the other one is not? That's of course because of epigenetic patterns, gene regulatory patterns that will activate or deactivate one of the alleles. And I have shown here on the slide the many options that have already been described for different genes. And sometimes it's even a combination of all those things. Can be differences in DNA methylation, the promoter that silences this allele, while here there is no DNA methylation. It can be enhancer competition, it can be binding of CTCF, I will talk about that in the next slide, which leads to some kind of boundary between elements, so that this enhancer here can only bind when this is methylated, while here it is blocked by CTCF binding to it. I will show in the next slide how that could happen. It can be antisense interference, so RNAi. At transcriptional level, we also see that small interfering RNAs can have an effect at transcript level, but also post-transcriptionally. That I will talk about in one of the, I think, in two classes where we are talking about long non-coding RNAs and short interfering RNAs. I will explain those things more. And then heterochromatinization, which will propagate until there is an insulated element, for example, and this can silence one of the alleles. If that allele is heterochromatin and the functional one is the euchromatin. So, various mechanisms. Let's give you one example. The IGF-2 region is extremely well studied, so I can explain you how it works there. But to be able to explain that to you, I need to tell you first something else. And that is those insulators or boundary elements that I talked about that separate between specific states in the chromatin, for example, euchromatin versus heterochromatin. They are very often present in the genome, very regularly at specific locations, so that they can be a boundary between chromatin regions. So insulator is synonym for boundary element. Can be between heterochromatin and euchromatin, but even between genes or groups of genes, they can act as kind of a separator, as a boundary. Now this boundary can be connected to something. This boundary can bind, well, a boundary protein can bind to the boundary element. And one very well-known example is CTCF. It's a boundary protein binding to a boundary element, but it's only binding when it's unmethylated. It's methylation sensitive. So only when the boundary element is not methylated, CTCF can bind. And CTCF connects with itself, with other CTCFs, let's say, not with itself, but with other same proteins, other CTCFs, and can make this kind of connections creating loops in the chromatin. So if CTCF, that CTCF here, is bound to those red things which are the insulated elements, you get looping. And that looping also, of course, influences the interaction between regions in the chromatin that are now either isolated completely from each other or are now more closely connected to each other. So for example, the pink gene here is inactive because its enhancer element cannot interact with it. While this gene here is suddenly activated because due to the loop, this enhancer element is now very close to the transcriptional start site. So you can have a transcription factor binding here and activating RNA polymerase 2. And then sometimes it also has no influence, of course. Like here, it doesn't matter. They were already close together, and by the loop, that is not affected. So this insulator has, can connect or disconnect specific regions of the chromatin so that they can interact with each other and genes can be activated. Those loops lead to organization of a chromatin inside that nucleus. Those loops lead to things, to a structure, a 3D structure of a chromatin, so that some parts are connected to each other and others are not. And we call those things also topologically associated domains, so large genomic regions that are interacting with each other because they are closely connected due to the looping. So they are high order chromatin structure, 3D structure. Those structures are isolated from each other by those insulators or those bonding elements. And specific proteins bind to those insulated elements like CTCF. There are also other ones, coenzyme and nuclear lamins. Nuclear lamins are found here, just below the nuclear membrane. You have the nuclear lamina over here, consisting of lamins, proteins, and they can also bind to boundary elements, so that the chromatin here, for example, is connected to the lamina through interaction with the lamina. So I had to explain that so that I can help you to understand the IGF imprinting patterns that we see. Just one example of imprinting also when it's when there is a mistake in the imprinting, when the patterns are not correct. You get suddenly biallelic expression instead of monoallelic expression, or the wrong allele is expressed. Then you can have these kind of weird inheritance profiles of certain diseases, like seen here. A maternally expressed gene, while the paternal gene is imprinted, then you can have a transmission of disease only through the mother. This mother here is a carrier. Only when the mother is carrier, her children can be affected. But if the father is carrier, you will not have that. The children are not diseased. Some of the children do have it's in heterozygous form, but are not diseased, so they are only carriers. So these kind of weird, let's say inheritance patterns of certain diseases are typically associated with mistakes in imprinting. So let's go to IGF-2. That's the best studied example of imprinting in humans, in mice, in many mammals. And I will try to give you an overview of what it does and how this imprinting works. IGF-2 stands for insulin-like growth factor. If you have a homozygous mutant, so that has been done in mice, homozygous mutants were made in IGF-2, and you see a strong reduction in growth. So, meaning that IGF-2 is important for growth of an organism already from embryonic stage, but also throughout life, you see that reduction, continuous reduction if you're homozygous mutant for IGF. But under normal, undiseased conditions, you have maternal expression of IGF. While the maternal, as I said, maternal genes tend to counteract the paternally expressed genes. Well, you have in HL maternal expression of an IGF-2 binding protein that we call IGF-2R. So it negatively influences IGF-2 to keep it under control, to say, whoa, 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 not too fast, don't grow too fast. I also need to survive as one, because IGF-2 is creating overgrowth of an embryo and later, of course, also of the placenta, excuse me, and then later also promoting, of course, growth of them. And H19 is also maternally. So if you have a heterozygous mutant now, two different possibilities occur. So the homozygous mutant, in general, you have 40% reduction in growth. But for the mutant IGF-2 allele, if you get it from your mother, that's no big deal. You will see a normal sized mouse developing. Even if there is an IGF-2 allele, because it's not active anyway. So it doesn't matter if it is mutated, a mutated allele that you knock out, you still get a normal mouse developing. However, if you get a mutant IGF-2 allele from your father, you will see, your father, the mouse's father, you will see a dwarf mouse. You will see the mutant growing extremely small. Because you normally should have high expression of that allele for normal growth, but if it's mutated, that will not happen. How come that we have this really paternal expression? Quite complex. This is the IGF-2 locus. The IGF-2 locus, you see IGF-2 here. Close by you have some other genes and elements. Like imprinting control region 1 is here, ICR1. Then you have a gene here called H19. Here you have a few enhancer elements. A bit further downstream, that's why you have these two lines here. It means a little bit further. It's not just next to it, a little bit further away in the genome, but still on the same chromosome. You have a gene called KCNQ1. Who invented that name? So if you ever have to imagine a name for a gene that you're studying, please do not call it KCNQ1. No one can memorize that. No. And there is an antisense. There is at the other strand, there's another coding region, which is called KCNQ1OT1. I will say KCN to make it a bit simple, KCN and the antisense of KCN. Because this here is a coding gene, but here this is not coding for a protein. It will code for an RNA that is antisense to the other one, and so can inhibit expression of the other one. Okay, and then here you have CDKN1. CDKN1 is cell cycle regulating. Okay, so these things are close to each other. Can interact with each other because they are in the same loop and the same chromatin. They are not in the same loop. It depends on the state. Excuse me, but they are closely connected, so they could potentially interact with each other. On the maternal allele, ICR1, it's a boundary element or an insulator element. Those are synonyms. Can be bound to CTCF because it's unmethylated. Okay, so ICR1 of the maternal allele is unmethylated, so CTCF can bind here. CTCF binds here and makes a boundary between the right side and the left side. So in the maternal allele, those things are not connected. They are in different domains and different chromatin loops. So these enhancer elements cannot bind to anything over there. So these enhancer elements, so I took the transcription factor that binds to the enhancer elements, can only control things within their same loop. So within the same loop, there's only H19. So H19 is active. But what is here? IGF2 is inactive. Then a little bit further here, you have KCNQ1 and OT1. Here, this one is active, and that one is inactive. It has nothing to do with this ICR region here. This ICR region here only has an influence up to there. ICR2 here is controlling what happens here. There's another imprinting control region, ICR2, that is in the maternal allele methylated. This ICR2 suppresses expression of this unsense RNA, making this one active. And this one as well is active on the maternal allele, CDKN1. But on the paternal allele, you see the inverse. The ICR1 is methylated on the paternal allele, and ICR2 is unmethylated here, leading to exactly the inverse expression patterns, of course. These enhancer elements are now closely connected to IGF2 because CTCF does not make a boundary. So the enhancer elements can activate IGF2 and do no longer activate H19. They are through the chromatin loops closer connected to each other so that IGF2 is now being. Because ICR2 here is unmethylated, you get expression of this antisense or suppressor RNA. This suppressor RNA blocks the expression of KCN and of CDKN1. So, that was on the next slide, the paternal allele, ICR1 is methylated and ICR2 is unmethylated. So the methylation pattern of the imprinting control regions determines which genes are active at specific locus. And that leads to those specific weird expression patterns where you see completely the opposite between the maternal and the paternal allele. So from the maternal allele, H19, KCN, and CDKN1 are expressed, while from the paternal allele, IGF2 and the antisense, that's this one is not expressed, is the antisense, that is the KCNQ1. Yes.
Does the binding of the CTCF cause the loops, or does the not binding of CTCF cause the loops?
The CTTF binding puts a boundary. It causes the looping indeed. Yeah. So it leads to, but the not binding will lead to other loops. You understand? If you remove it, still other ones are. So the looping will just change by if it binds or it doesn't bind. Yeah.
I was confused that in the maternal allele, CTCF binds to ICO1.
Let's go to the maternal allele.
Now, because of the looping, I would believe the enhancer elements will be closer to IGF2. Because in this picture, they're not looped, right? This is just the normal conformation, the enhancer elements.
Yeah, they don't show the looping. That's the annoying thing. They don't show the looping, but these two, are separated from each other by this looping. So they are in different parts. So this is in one loop and this is in another loop. And that is why these enhancer elements cannot reach IGF2.
And when CTCF is loose, then they're closer to each other.
Then they are in the same loop. Yes, indeed. Yeah. So the chromatin looping is not really clearly shown here. Yeah, I must say I find it very difficult to find a clear representation of these. I have been looking for other graphs, and this is the best one I could find. They don't show the chromatin loops, and that makes it a bit hard. I understand that. But trying to draw it myself with all these things, I decided to take this one, but indeed the looping can be a confusing part. So as long as you know that the boundary elements
I think they
are
try to
differentially methylated, yes?
they try to show it with the arrows, right? The full lines, like the ones being close to each other.
You mean this one?
Yeah.
Yeah. Yeah, that's what they want to show indeed, that you cannot access that other part. Or the enhancing elements cannot go further than this because there is an isolation, a boundary element here. Disconnecting them from each other, separating them in different chromosomes. Now that, and now I'm going to make it even more complicated. IGF2 and H19 are on this locus here on the chromosome, but there is also something, and I said, there's also IGF2R. What is that then? IGF2R is the negative regulator of IGF2, and it modulates how much IGF2 is present in the cell. And that is at another locus in the genome, at this location here that you see represented here. Again, the two alleles, the maternal and the paternal allele, have a completely different expression pattern. You see multiple genes here. The one that I'm going to focus on, IGF2R, and some other ones as well, again have a inverse almost expression, not completely inverse, but different expression pattern between mother and father. Not all of them have that, but IGF2R does. So in the maternal allele, IGF2R is active, and there is no expression of a non-coding RNA called AIR. However, on the paternal allele, this one is inactive and AIR is active, and that's a non-coding RNA. And we see a lot of differences here in histone modifications. So nothing related to DNA methylation now, but here histone modifications are allele specific, are different between the maternal and the paternal allele. There is a little bit of differential DNA methylation, but not so much. It's mainly the histone modifications that are strongly different between the two. And you see different expression patterns here on the IGF2R, here mainly AIR, and then some other genes close by are biallelically expressed. So it's not certainly not for all genes on this locus that you see this monoallelic expression pattern. So these are things that are best studied, and still we don't know everything about it. So you could ask the question, when and how do these marks get set there? And that happens early during embryogenesis. We will talk about that later, that you get the epigenome being set, but it's not so clear what determines the difference between the paternal and the maternal allele. We just observe that this happens, that this is going on in embryonic development, that suddenly you see those differences. So, but who has put them there? What drives these differences? It's not right. So what then explains the difference between the liger and the tiger? Well, it appears that imprinting is extremely strong in lions, but in tigers, not. In tigers, IGF2, this is gene IGF2, is biallelically expressed. Female or male tigers, it doesn't matter, the gene is moderately expressed. So a normal tiger has a little bit of expression from one allele of IGF2 and a little bit of expression of the second one, and that is fine. However, in lions, this pattern of monoallelic expression is extremely strong. In lions, the IGF2 gene is on the paternal allele, and it's off on the maternal allele. in a regular cross between a male lion and a female lion, that is fine. You get one allele active, very active, and one allele off. That's good enough. The lion will normally grow. However, if you now make a cross between those things, you will see that you disturb the whole pattern. And so if the father is a lion, you have extreme strong expression of IGF-2. And that leads to this huge offspring, the huge child that we saw there. So if the offspring grows enormously big if the father is a lion. Another typical imprinting phenomenon is X chromosome inactivation. I think you all know that females have 2 X chromosomes, but not both of them are active. One of the 2 is inactivated in every single cell of our body, except for the oocyte, where they are again reactivated, of course, after just after meiosis. Random inactivation. So it's not that every cell in our body has the same inactivation, and that is because it starts this inactivation. So the moment that one of the 2 X chromosomes gets inactivated starts when the embryo, the female embryo already has 2 to 8 cells. And it takes about 8 days. So it's during the blastocyst stage that we get this inactivation, and it's random for every cell. So we are all mosaics. We are all chimeric as human beings. As females, not as human beings, as females, meaning that in every single cell it can be, in the various cells it can be different X chromosome that is inactivated. This inactivation, again, typical patterns that we see there, DNA methylation all over the chromosome. Alternative histones bind to it, macro-H21 is another histone. Instead of the regular H2A, macro H2A. I said H21, H2A, macro H2A. On this inactive chromosome, it is then transmitted to the daughter cells. So if in one cell it's inactivated, the daughter cells have the same inactivation pattern. And during meiosis, of course, it needs to be reactivated. It would be really annoying if you would get from your mother an inactive X chromosome. So certainly as a boy, you need that active X chromosome from your mother. So in all the oocytes, the X chromosome is again active. And it's not complete. We see that the X chromosome is 90% inactive. 10% of the genes still remain active. Now you can clearly see even that inactive X chromosome, if you stain such a, like for example here, a human female fibroblast is stained with DAPI, which stains DNA, and on the right side, even with an antibody against this macro H2A, then you clearly see something here, very bright, but here it's even more clear, of course, with the enrichment of macro H2A in something that's inactive X chromosome. We have called that the Barr body, because it was discovered by a certain guy named Barr, who observed it. At that moment we didn't know yet what it was, of course. Later only it was discovered that that is the inactive X chromosome. Macro H2A does not only occur in the Barr body. You see some dots at other locations as well, but it's enriched over the place. And why is that? Why does one of those X chromosomes get inactivated? Well, we see that the number of copies of each chromosome is very important. So you probably all know the Down syndrome, where there are 3 chromosomes 21, and that leads to problems with the normal development because of gene dosage effects. It's really important that you have the correct number of genes so that you have a normal development. this disequilibrium can cause some of these developmental abnormalities, let's call them, like Down syndrome, also Turner syndrome, where that is caused by having only one X chromosome, is also a disorder that is related to the disequilibrium of the chromosome numbers. And it can vary in your body, and that leads to those cats, for example, or mice as well, with lucky spots with different patches on their fur, different colors of their fur, caused by a gene encoding color, so an enzyme that is responsible for color formation, being inactivated in some parts of the genome, of the organism, and not in other parts, and therefore you get this fur that has different colors. You only see that in female cats, of course. So the Locust cat, so this cat, this female cat. You also have a white part there, so the X chromosome inactivation explains the dark and light brown parts. The white part that you see there, that's another gene. So that's even suppressing any color formation in some parts of the body. And that is mainly activated by other mechanisms than in eating. So I think you also see here that this cat even has different eyes. The eye color varies a little bit. Also in humans it has been described that color blindness is located, the gene responsible for that is located on the X chromosome, and some females have one eye that can see color and another eye that is colorblind. So even it can vary between the eyes. It's quite exceptional, but and that's because females are chimeras. So if you accidentally have complete eye with that chromosome inactivated, you are colorblind only on that eye. Of course, your other eye will come. So it's a process, this inactivation. It's not black and white. It's not that suddenly the chromosome is inactivated completely. It happens gradually during embryonic development. It starts somewhere between the 2 to 8 cell stage of the embryonic development. You see here suddenly initiation. At a certain moment, one of the X chromosomes is starting to produce cysts. And again, we don't know what determines which of the two will express cyst. You get expression of cysts here on one of the two X chromosomes. And in each cell, it could be another one of these 8 cells. When it starts, cyst will cover XI, inactive X, while the active X, XA, the active X, will express an antisense to cyst. It will express the opposite version, so 6, TSIX, to protect this chromosome and to keep that chromosome active. 6. So it all starts from that inactivation center here, somewhere on the inactive X. Cyst is going to be expressed, and cyst A is located at the X chromosome inactivation center. It's a quite long non-coding RNA. It doesn't code for the protein. 70 kilobases long. And it works in cis, meaning it will bind to close by genomic region, the same genomic region, to that X chromosome. It covers it. It will kind of condense it. It's essential that this happens, but that's not enough. More things are going to happen for full inactivation. What else do we see on that inactive X chromosome? Histone modifications. The histones are deacetylated. We see a lot of H3 lysine 27 3 methylation and a lot of DNA methylation. something that is antisense to CIS, to interfere with CIS, so that CIS cannot bind to it. So that CIS, if it would be close by, that this active X chromosome remains protected from CIS binding. So this happens, it's first initiated, then established, and then maintained when you have the gastrulation. And we see here, when those, during that stage of the embryonic development, during gastrulation, it's finished. But here, going from here to there, you go from 8 cells to about 200-300 cells. So, in all those cells, it could vary. It could be that another X chromosome is inactive, and that explains that chimeric observation that we have in the females. So by the time of the gastrulation, it's finished. Now, a lot of disorders that we have observed over the past 50 years could initially not be explained. We saw these weird inheritance patterns that are now found out to be linked to imprinting mistakes. So if accidentally IGF2 is expressed from both the maternal and the paternal allele, you get a syndrome called Beckman-Wiedemann syndrome, and we see a lot of overgrowth of the placenta, high birth weight, and many other observations that are shown here on this slide. Another well-described imprinting disorder, the Angelman and the Prader-Willi syndrome. I did not explain those imprinted genes, but it's a part on chromosome 15, and it's the, what was the name again? It's some UBC gene, if I remember it well, that needs to be expressed from the paternal allele, and if that is not happening, you get either the Prader-Willi syndrome or the Angelman syndrome, depending on UBE3, that's the name. It's in here. Depending on whether you get it from your mother or your father, you have a different set of symptoms that you can observe in those children. And also Turner syndrome is linked to the dosage disequilibrium. People with Turner syndrome have only one X, females with only one X chromosome. And this is a very famous actress that has Turner. She's quite small in size, but it shows that even people with Turner syndrome can be can have a very good life, and her name is Linda Hunt, and also be very social and have a nice career as well. So it doesn't necessarily mean because you have this linkage disequilibrium that everything is wrong, but it depends. Because we see variation depending on whether you get your maternal X chromosome or whether you get the paternal X chromosome. As in the Turner syndrome, whether you have only the one from your father or only the one from your mother remaining. So this population of people with the Turner syndrome are very often studied to investigate imprinting on that chromosome. And by doing so, we have discovered that X chromosome imprinting is often having effects on social competences, on body growth, on body fat and cholesterol. Things that are typically different between females and males. And could potentially add, so X chromosome inactivation could potentially, and the imprinting patterns that we see there, could potentially even explain why females and males are different. And I'm talking about biological, females and males. I'm not talking about gender. That's something. And yet another population that we like to study in epigenetic studies are the twins. Because the twins are also extremely interesting in terms of genetics versus epigenetics. genetically they are identical. And I'm talking about monozygous twins, they are identical. But over time, the older they get, the more different they are. Genetically, they are of course still identical, but epigenetically, they are different. So the older they get, the more you see the effects of the epigenetics. The more you see that the environment has an influence, has interacted with that genome, and leads to different phenotypes. And these 61-year-old ladies here had a different lifestyle. They are monozygous twin, but one of them smoked for many years and liked to be in the sun, and the other one not. And I was going to say, which one is which? I think you can see. So they have, if you compare young twins with old twins, so a population of young twins with a population of old monozygous twins, you will see that their genetic makeup, of course, is the same, but their epigenetic profile starts to gradually change the older they get. And we see clear differences between DNA methylation profiles, not only that, but also between histone modification profiles. Here is shown. The difference in histone acetylation is somewhere shown. This is DNA methylation. So 3-year-old twins in general have about 2.5% of their genome, of their Cs in their genome are methylated. For the 15-year-old twins that were studied here, you see a big difference between 3.5 and 4.5. Same. What you also see here, that in general, over life, we get more and more DNA methylation, all of us. 3-year-old twins, acetylation, almost same level. Histone acetylation at age 4, around almost 50%. And then older, you have again this gap. One of the two has around 50%, and the other one is 65% histone acetylation at age 4, and this is here for age 3. There you see the drop over time, and again, difference between the two individuals of the twin. And the more these people have a different lifestyle or have experienced different things, the more changes you see, the more differences you see. Yeah, this is what I already said. The epigenome changes with aging, not only with that, also influenced by the environment. And here, this is a technique with which you study methylation using methylation-sensitive restriction enzymes. It's called AIMS. It's a quite old technique, so that is why I didn't have to explain it. But I quite like the fact that you see here very clearly the difference between the banding patterns, so the kind of fingerprints, the epigenetic fingerprint of these 3-year-olds is very similar, while here for these 15-year-old twins it's completely different. And they looked how many of these differences they could observe, and in 3-year-old there were only 5 bands, so this is just a few that I show here, it's only a subset. They monitored more than 100 different of these fragments with methylation-sensitive restriction enzymes, and here only 5 were different between 3-year-old twins, and at 50 years, 70 of them were different. So, SNPs in the genome, epigenetic marks are together, of course, explaining different phenotypes. So our SNPs, our genetic diversity, our genotype explains about 20% of the differences we have calculated. Only 20% of them can explain differences in phenotype, but the rest is mainly related to epigenetics. So phenotypic differences between all of us here, yes, it's based on genetic differences, but it's largely based on epigenetic differences between us. So the epigenome is like almost 80% responsible. This is just, these are calculations, of course, estimates. So don't pinpoint me on those numbers. It's just so that you have an idea how important that epigenome is. to have more information about what I have discussed today, chapter 6 and 7 of the book, that is interesting to read. And certainly do not study again for chapter 7 this part. Okay, that is the end for today. Maybe for people that prefer to read a bit more about Lykos and